Um, Welcome to this deep dive. Today we're unpacking a botanical source document titled Erter.
Right, which explores the science of growing peas. And our lens for this is through two nature and innovation students, Kesser and Steen.
Yeah, they're basically prepping old seeds for next season in Steen's family kitchen garden.
And you'll see this isn't just about reading a seed packet. It's really an exploration of creative scientific inquiry,
Hmm.
sustainable innovation, and, well, the hilarious reality of forgetting your own research.
It is a pretty funny setup. I
Right.
mean, they're testing these old, dry pea seeds from Tantigran and Green.
Yeah.
But Kesser brings up this massive botanical experiment they did just last spring, and Steen has completely forgotten it.
He says his brain is as blank as stainless steel.
Which is wild because he literally wrote the report and presented it to the class.
Right. So he realizes he needs more what he calls dirty duty.
Getting soil under his fingernails.
Exactly. To truly absorb the gardening knowledge, because passive learning it didn't stick.
And it's ironic because that forgotten experiment was actually incredibly rigorous.
Oh, highly rigorous. It was a 4-group control study.
They were comparing standard rainwater against salicylic acid.
Yeah. And not just any acid. They derived it from a spontaneously fermenting bokashi-style willow bark soak.
Which is just a huge amount of effort. And they didn't stop there. They even planted the seeds in Fibonacci spirals.
And concentric circles.
Right, in these really large pots.
All of that geometry was just to optimize light exposure and give the roots enough space so they wouldn't compete.
But because his mind is a complete blank, their current plan is way simpler.
Going back to basics.
Yeah, just soaking the peas to rehydrate them, which is actually a trick from Oma to easily spot the best, most viable seeds.
Right, before they plant them in recycled paper towel plugs. Which brings in some really fascinating biology.
Oh, the root systems.
Yes. Pea taproots can dive 25 to 40 centimeters deep.
That is massive for a little pea plant.
It really is. Therefore, they need planters with 30 to 40 centimeters of soil depth just to accommodate that.
So a standard balcony box won't cut it.
Not at all. And above ground, these specific plants will grow 45 to 60 centimeters tall and 25 centimeters wide.
They demand a lot of space. With those requirements, I know Steen was super curious about hydroponics.
He was, though an AI warned them that peas absolutely hate having their feet in water for long.
They are so highly adapted to soil ecosystems.
Which leads to this ultimate aha moment for Steen. He remembers asking Professor Hendricks if peas really pull nitrogen from the atmosphere.
And they do.
They absolutely do. They utilize this fascinating collaboration with beneficial bacteria on their roots.
It's basically an invisible partnership.
Exactly. The bacteria convert atmospheric nitrogen into a usable nutrient for the plant.
So for you listening, whether you're gardening or just learning a completely new skill, Steen's realization is a perfect reminder.
Getting your hands dirty matters.
Yeah, that hands-on dirty duty makes complex information stick far better than simply reading instructions.
It really does.
Right.
And it leaves you with a pretty profound final thought to ponder.
Let's hear it.
If pea plants are quietly using microscopic root bacteria to pull nitrogen directly out of thin air,
Which is amazing on its own.
right, what other invisible, complex partnerships are happening right beneath our feet in the soil that we completely take for granted? We're jumping straight into something really cool today. A hands-on experiment by two young students, Kesser and Steen.
Yeah, it's fascinating. They're looking into plant growth.
Specifically using a natural approach, right, salicylic acid.
Exactly, from willow bark, seeing if it gives pea plants a boost.
Okay.
And we've got some great source material excerpts like Erter 1 Sow Experiment Med SA, which is basically Peas 1 Sowing Experiment with Salicylic Acid,
Right.
and another called Placering Ipotin Placement in the Pot.
So we get a real peek behind the curtain at their planning.
We really do. It shows how thoughtful they're being as they set this all up.
So let's get into it. Kesser and Steen.
Yeah.
They have a clear goal here, don't they?
Oh, yeah. They want to see exactly how this willow extract affects their peas.
Everything from like germination.
Germination, growth, flowering, the final weight,
Wow. Okay.
and even secondary things, like taste, how long the peas last.
But even next season's sprouting, that's pretty comprehensive.
It really is. They're not just looking at size, it's the whole cycle.
And their inspiration is this mix of science class and Oma's gardening.
Yeah, their grandmother. Apparently, she always soaks her pea seeds.
Why does she do that?
She thinks it helps her see which seeds are the good ones before planting.
Ah, interesting. And that ties into their first step.
Directly. They soaked their seeds too, but in two different things.
Okay, what were they?
Plain rainwater, that's the control group.
Makes sense.
And then this willow water they made themselves
How do they make that?
by soaking fresh willow cuttings in rainwater. Some they peeled, some they just chopped up.
Okay.
And they actually noted something interesting. They called it Peelevand med naturligering.
Natural fermentation in the willow water.
Yeah, which, could mean other compounds are in there besides just salicylic acid.
Huh. So that adds another layer, potentially?
It does. It's a variable. But the fact they observed it and wrote it down, that's great scientific practice for young researchers.
Yeah. That is impressive. Shows real attention to detail.
It really does. Real-world experiments often have this kind of, complexity.
Okay, so seeds are soaked. Now, planting them in pots.
Right. They got some fairly large pots.
From the man in the green shirt. Sounds like a local character.
Apparently so, at the garden center.
But they started with, what, 15 seeds?
Yeah, initially. And they were thinking maybe rows, like 2 rows of 5.
But then they started discussing.
Should they stick to maybe 10 plants for each group? The rainwater ones and the willow water ones.
And keep some spares.
Exactly, just in case some don't germinate, or for other observations.
That's smart planning. Thinking ahead about potential problems.
Definitely. It shows they're adapting, not just following steps blindly.
So how do you arrange 10 plants in a round pot? That sounds like a puzzle.
It is, and they really thought about it. The pot's about 40 centimeters across.
Okay.
First idea, rows, maybe a 3-4-3 pattern.
Simple enough?
Then concentric circles, like a bullseye.
A bullseye. How would that work?
1 in the very middle, then maybe 3 in a ring around it, and 6 in the outer ring.
Ah, I see.
And the clever part there was thinking they could put, 5 assay-treated seeds and 5 rainwater ones in the same pot.
Oh, to keep conditions similar for both groups.
Exactly, trying to control those variables within the same pot.
But Kesser had another idea. Something about Fibonacci.
Yeah, like a snail pattern.
A snail pattern. Why?
The idea was to get really even exposure for all the plants. They were aiming for about 5-7 centimeters spacing between them.
Wow, Fibonacci. That pattern shows up in nature for efficient spacing, right? Like sunflowers.
Precisely. So Kesser was tapping into that idea for evenness. It needed visual dots, apparently.
Meanwhile, Steen's grappling with this.
Yeah, he had a bit of a headache thinking about it. But then inspiration struck.
From what?
His dad's spiral fairy lights.
No way. Fairy lights?
Yep. He even thought about using a knotted string inspired by the lights to measure out the spiral pattern.
That's fantastic. Finding solutions in everyday things.
It really is. Shows that kind of creative observational thinking.
And then Kesser circled back with another idea.
Possibly sparked by Uma again. Planting cuttings along the very edge of the pot.
Along the periphery. What's the thinking there?
Better root growth, maybe? And it also leaves the middle clear.
Clear for what?
For support, like chicken wire netting for the peas to climb.
Trellis.
Yeah.
And protect from birds, too, maybe.
Right, and also space for a measuring stick right in the center to track height easily.
Okay, so all these ideas are kind of merging into a plan.
Exactly. They're thinking planting, support, and monitoring all at once.
Now, about the plants themselves, they did some homework.
They did. Found out pea roots can go pretty deep, up to 40 centimeters.
Which explains why they needed those larger pots.
Definitely justifies it. But then they saw other people growing pea shoots differently.
How so?
In really shallow trays, or even gutters, just using flower water.
Huh. That's quite different.
It made Steen wonder about hydroponics.
Especially since they read peas don't like staying too wet. Their wet feed issue.
Exactly. So they're comparing methods, thinking critically, not just accepting one way.
That's good. And they considered the plant size, too.
Yeah, the variety they chose gets about 45 to 60 centimeters tall, maybe 25 centimeters wide.
So 10 plants in one pot might get a bit crowded.
They acknowledge that,
Yeah.
potential crowding. But they also noted, people often plant peas close together in rows in gardens.
True. So they're balancing ideal spacing with practical reality.
Seems like it. Grounding it in how things are often done.
Okay, let's talk experimental design. How did they actually structure the groups?
Well, the source material suggested maybe 5 or 6 seeds per group, good for observation, plus backups.
Right.
And Kesser and Steen came up with a really well-structured plan, a 2x2 design.
Okay, break that down for us.
Sure. You've got 4 distinct groups. Group A seeds soaked in water, and they get watered with water.
Simple control.
Group B soaked in water, but watered with the willow water. Group C soaked in the willow water, but watered with plain water.
Got it.
And group D soaked in willow water and watered with willow water.
Ah, so you can isolate the effect of soaking versus watering and see if they combine.
Exactly. It's a factorial design. Super useful for seeing not just individual effects, but also potential interactions between the treatments.
That's really quite sophisticated planning.
It really is. Shows they're thinking systematically.
And they knew they had to track progress carefully.
Oh, yes. Planning to keep a diary?
Noting down.
Dates, plant height, number of leaves, when flowers appear. The whole progression over maybe 3 or 4 weeks.
Good data collection.
Plus, a great idea for visual documentation, weekly photo.
With different colors for each group.
Yeah, using color markers to identify group A, B, C, and D in the pictures.
That'll make comparing them really easy.
Definitely creates a strong visual record alongside the numbers.
And one more detail using paper tubes.
Right, for starting the seeds. Pre-sprouting.
Why do that?
Peas can be a bit fussy about having their roots disturbed during transplanting.
Ah, okay.
So starting them in paper tubes, which you can plant directly in the pot and the pepper just breaks down, helps reduce that stress.
Clever. Minimizes root disturbance.
Exactly. Helping the seedlings get established well.
You know, listening to all this, what really stands out is just their curiosity, their approach.
Absolutely. It's hands-on, inquisitive.
Yeah.
It might look a bit messy compared to a formal lab, but it's genuine science.
They're thinking independently, building their own system from observation.
And you see connections to bigger ideas, don't you?
Yeah, like sustainable agriculture, using natural resources.
They're exploring chemical-free ways, drawing on OMA's knowledge and their own observations.
It feels very bottom-up.
It is. Contrasting with just being told, do this. They're figuring it out, trusting natural methods.
And they're okay with not knowing everything up front.
Right. Seeing the peas as indicator plants.
That attitude of, we don't know yet, but we'll find out.
That's the core of discovery, isn't it?
And they actually want you, the listener, to get involved.
Yeah, they created a hashtag how to grow pea shoots.
So they're inviting you to try your own pea experiments and share them.
Which is brilliant, fostering that shared exploration.
Definitely. And we really encourage you to look at the notes for this deep dive. There might be visuals, more details on their planting patterns and setup.
For sure. And hit subscribe so you catch our future deep dives.
Okay, so let's just quickly recap. We've walked through Kesser and Steen's really careful process.
From soaking the seeds in that homemade willow water,
Through all their thinking about planting patterns in the pots.
to their structured experimental design and documentation plans.
It's genuinely impressive.
And the real aha for me is just seeing how that focused curiosity, that careful observation,
Can make even a simple setup like growing peas reveal really interesting things.
it shows the power of just looking closely and asking questions.
Which brings us to our final thought for you to chew on. How often do we just walk past potential discoveries in everyday stuff?
Yeah, think about it. What other simple natural processes are out there?
What could we learn if we just applied that same kind of dedicated observation that Kesser and Steen are showing?
Makes you wonder, doesn't it? What else is waiting to be understood right under our noses?